Hi everyone, this is the Immunology Applications lecture. In this lecture, we're going to learn about vaccines and their mechanism of action, and then we're going to talk about immunoassays, which are different tests you can do in lab using antigens and antibodies to determine if someone has an infection. We can use them for various things health-wise. So we're going to do a quick outline here. Let me start. So we're going to talk about different categories of vaccines, and some of you have been hearing about herd immunity with all the coronavirus stuff going on. So we'll define that, and then I'm going to talk about just different immunological testing that's very popular in different companies that we do to determine if someone has an infection, pregnancy tests, HIV tests, and then we're going to end with what monoclonal antibody therapy is. So just to talk about immunization again, because we finished talking about the immune system, immunization is when someone is made immune to something, to an infection. Immune means you're resistant to it, so you're not going to get it again. And you can have natural or artificial immunity. Naturally acquired immunity is exactly what the name says. It means it's acquired naturally in your lifetime. So naturally you getting an infection and being immune to something, or naturally by getting antibodies when a woman is pregnant, so the baby getting antibodies, and as well as from breastfeeding. Artificially acquired immunity is when you get immunity not naturally, so artificially. So you actually take something to boost your immune system. That's an example of vaccination, and also sometimes we use antibody therapy with individuals who are sick, where we artificially give them antibodies so their body can fight an infection. Then there's active immunity and passive immunity. Active immunity is when your body has an immunity from exposure to an antigen naturally or artificially. So it's, I guess, for the best way to describe it is it's your body actively fighting an infection and then producing an immune response. Passive immunity is when you get immunity when you become resistant to an infection passively, by basically not doing anything yourself. So with active immunity, you can have active natural immunity. That's just by you getting an infection in your lifetime. So if you've had a cold, you now have an active natural immunity to that strain of virus that caused a common cold in you. Artificial active immunity is what vaccination does. So it gives you immunity by getting artificial injections of pathogens or portions of pathogens. And then passive immunity, you can naturally get it from a mom either when she's pregnant, antibodies are passed to the fetus, or during breastfeeding, antibodies are passed in breast milk. And then artificial passive immunity is when you give a patient antibodies. So those are different types of immunization. So here there's active natural immunity, artificial active immunity, which is vaccination. That's what we're going to focus on. Natural passive immunity, artificial passive immunity. And we're going to focus on vaccines right now, which are an example of active artificial or artificial active immunity. So immunization, you being resistant to a disease, can be achieved by actually getting a vaccine. And what the definition of a vaccine is a suspension of a pathogen. Remember, a pathogen is anything that causes disease or components of a pathogen. So look at this shot right here, but just remember, not all vaccines are given as injections. Some vaccines are nasal, some vaccines are given orally. Anyway, whatever that component is whether that's orally or injected, it's a suspension of the pathogen or components of that pathogen. And when we inject it, or the person takes it, however they take that vaccine, it induces active adaptive immunity, making the person have an immune system to that pathogen if they ever are exposed to it. The vaccines are composed of inactive or harmless versions of a pathogen. They're not actually the pathogen that's going to make you sick, because if we were doing that, then we would be getting people sick. So vaccines contain weakened or dead pathogens. And It is really the only way we have to deal with deadly viral diseases. Viral diseases are not that easy to treat. So bacterial diseases can be treated with antibiotics. Remember, bacteria and viruses are completely different microbes. We cannot treat any viral diseases with antibiotics because antibiotics just target bacteria. With viruses, antiviral drugs have not been that effective. So a lot of times we give vaccines for certain virus infections that can be deadly, and this is how we've prevented them. This is also what we're doing right now with coronavirus, is trying to produce a vaccine that will target the virus so that we don't see any more disease COVID-19 cases. The way a vaccine works is it's injected. It's not really injected because, again, there's vaccines that are oral. They're not that common, but there are vaccines that are oral or through nasal. And so it's put in you somehow, usually through an injection, and it induces an immune response, which causes your body to create antibodies and memory cells. The key here is memory cells, meaning that if you get a vaccine for chickenpox, the virus that causes chickenpox, and you're exposed to the virus that causes chickenpox years down the road, your body already has formed memory cells to target that virus so that you don't get the disease. And so later natural exposure induces a secondary immune response. Remember, secondary immune responses are stronger and faster. So the pathogen is destroyed before it can even cause disease. And that's why we get vaccinated. And so the main idea behind vaccination is to build herd immunity. There's many people who cannot be vaccinated due to many reasons. Maybe you're immunocompromised, you have certain disease, you're of a certain age, babies cannot be vaccinated for everything. So there's many reasons why someone cannot be vaccinated due to health reasons. So they're protected by everyone else who's vaccinated, and we call this herd immunity. So it's herd immunity is community protection, and it's when most people, and I'll talk about the percentage of people, are immune to a specific disease, so it's a specific viral or bacterial disease, so that the people who are not vaccinated are protected because we all are protected anyway. So this is why we have state-mandated vaccinations for children to build herd immunity. And according to the CDC, so the Center of Disease Control, herd immunity fails when 5% of a community is not vaccinated. So for herd immunity to work, you need at least 95% of a community to be vaccinated so that we can protect the 5% of immunocompromised or old people or pregnant women cannot get certain vaccines. So we need most of the population, 95% of the population, to be vaccinated or immune to something so that the 5% who are vulnerable are protected by the herd or the community. And again, it's immunity in most of the population. And actually, I want to stress something. So this lecture is about vaccines. So I'm talking about vaccines helping accomplish herd immunity. But okay, for example, if there were no vaccines and if we use like coronavirus as an example right now, there's no vaccine right now that works for SARS-CoV-2. They're trying to create one. But let's say most of us got the infection and survived. The key is that you survived, so you built immunity. So if 95% of the US, 90 to 95% of the US had the infection, didn't know, overcame it, now has the antibodies, and we were to go out, like if the quarantine was over, herd immunity would have been established so that we, the 95%, protect the 5% who are not immunized against or who have not had the virus. So that's how herd immunity builds. It's either through people actually getting sick and surviving. That's the key. Surviving the disease where most of the population gets sick, or where most of the population gets vaccinated so that we protect those who cannot get sick, cannot get vaccinated. And again, we need a critical amount of the population to be immune to something to build herd immunity. And this is the example, I used to use this example a lot with the class when I was pregnant. So if the whole class, for example, took the flu vaccine and none of them had the flu because they were all vaccinated and the vaccine worked with the students who took it, and I couldn't get vaccinated because of whatever reason. Maybe I had a certain disease or disorder or a pregnancy that prevented me from getting a certain vaccine. Well, I won't get sick because most of the people in my class are not sick, so I'm not going to get exposed to it. So now think about it in terms of a deadly disease. If most of the people are immune from a deadly disease, whether that's naturally because they got sick and overcame it or through vaccine, that's how we get herd immunity. And so vaccines have been very effective. They have eliminated many diseases, specifically viral diseases, from various countries. So when we think of polio, polio has been very, very, very low amounts because of the vaccine. And some diseases have been totally eradicated. Eradicated means gone because of vaccination. One example of such viral disease is smallpox. So smallpox is a deadly viral disease. And since we've created the vaccine, we have not seen any more cases of smallpox. So it was eradicated around 1977, 1980. And so the more people who are vaccinated against the disease, again, the safer it is for everyone because of herd immunity. And vaccines have really played a big part of this. So just to talk about some diseases that have been almost completely eradicated or totally eradicated by vaccines. Smallpox has been eradicated. These are the diseases that are almost gone due to vaccination. So diphtheria causes it's a bad infection of the upper respiratory tract. Pertussis, which is whooping cough, is nearly gone. Tetanus causes locking of joints. Polio can cause paralysis. Measles, mumps, rubella, just many different viral diseases that we've eliminated due to vaccines. So we're going to talk about the polio vaccine and its effect. So polio, just to start with that, polio is a viral disease that can lead to paralysis, which obviously is a terrible thing. So it's almost been completely eradicated due to vaccination. We are still seeing a few, I don't know if the word would be outbreaks, but we're seeing a few cases in some countries coming back. But the polio is a very contagious disease caused by the polio virus. The virus attacks the nervous system, so cells in your nervous system, and paralysis can be a temporary symptom or develop into a permanent symptom for some people. So here are some pictures of what polio could cause. There's no cure for it. We don't have like an antiviral drug that we can give someone to polio, but it can be prevented by giving the polio vaccine. So here in the US, we all take the polio vaccine. There's two types of polio vaccine. There's Dr. Salk and Dr. Sabin both created their own versions of the polio vaccine. There's an inactivated polio virus vaccine. We're going to talk about what inactivated and attenuated and all these terms mean in a second. But Dr. Salk created a vaccine that is a dead polio virus, in essence, in an injection that is administered and has decreased polio cases. And then Dr. Sabin has the oral, so you take it by mouth, polio virus, which is a live attenuated polio virus. This is not used in the US, but it's used throughout much of the world. And you can imagine that giving someone an oral vaccine is a lot more cost effective and needs less skills than giving someone an injection. So this is also one reason why this vaccine is more popular in other countries here. And when we look at the cases of polio over time, so when we look from 50 years ago over 50 years, we see that the virus cases have tremendously gone down with the vaccine. So we have Dr. Salk, who has the inactivated polio virus used in the US, and Sabin, which has the live attenuated polio virus. There's different types of vaccines, and we're going to talk about all of them. There's whole agent vaccines and fraction of the agent. When I say agent, I mean the bug, the pathogen, whatever you want to call it. So whole agent vaccines are vaccines that actually use the whole bug. So if you're looking at this picture that use the whole bug, they either weaken it or kill it. So those are whole agent vaccines to stimulate an immune response. All of this is to stimulate an immune response. Fractions of the bug or agent are when we take pieces of of the pathogen that caused disease and use it to stimulate an immune response. Maybe you would take the cell wall of bacteria or spikes of viruses. Spikes help viruses get in and you use them to mount an immune response. Or maybe it's not actually the agent or the pathogen that's causing the disease, but a toxin they produce. So you produce a toxoid vaccine. So we're going to talk about this. And we're going to talk about just some of the diseases. So with whole agent vaccines, again, there's live attenuated vaccines and killed inactivated vaccines. Live attenuated vaccines are like the MMR vaccine, which is measles, mumps, rubella, chickenpox vaccine caused by varicella virus, shingles vaccine caused by the herpes zoster virus. There's different types of influenza vaccines. Rotavirus vaccine, which for babies is given orally. Killed inactivated vaccines, examples of that are the Salk's polio vaccine, hepatitis A vaccine, and then another influenza vaccine. Toxoid vaccines is when we take inactivated toxins that would otherwise cause, so if you didn't have the vaccine and you got infected with organisms that produce the toxin, you would get sick. So we create inactivated toxin vaccines. That's like tetanus and diphtheria. Protein subunit vaccines is we take proteins from certain agents or pathogens and use them to create an immune response. That's pertussis, hepatitis B, the HPV vaccine, so human papillomavirus. And then polysaccharide vaccines is when we take sugars from different types of organisms, bacteria, and we produce vaccines. So pneumococcal pneumonia vaccine, meningococcal disease vaccine, and Haemophilus influenza type B meningitis vaccine. Okay, we're going to start with live attenuated vaccines. A live attenuated vaccine is a whole agent vaccine, so you're using a weakened pathogen. I'm going to use an injection in all of these because it's easier. If you could actually microscopically see in the components of that, there is a weakened pathogen. So you take the bug, the virus, the bacteria, and you make it weakened and you inject it in a person. So this is what live attenuated vaccine means. An attenuated vaccine is a live vaccine. The advantages is it mimics an actual infection, and a single dose can give you long-lasting adaptive immunity, so cellular and humoral immunity, because the microbe can actually replicate in the body. The disadvantages, because these vaccines are not very popular, is that sometimes, rarely, they can cause a disease in immunosuppressed individuals. That's why sometimes immunosuppressed individuals don't take certain vaccines, because they can cause the disease in them, because all of these are trying to mount a healthy, strong, proper immune response. They can rarely mutate in the body and change to cause the disease again. They're generally not recommended for pregnant women for these reasons, and they require refrigeration. The reason why this is a disadvantage is because you can imagine this is another layer of cost in keeping the vaccine working. So examples of live attenuated vaccines are the MMR vaccine, which has multiple components, the varicella vaccine, shingles vaccine, and rotavirus vaccines. These are all live attenuated vaccines. And a lot of times people are scared of these vaccines for these reasons, but I want you to know that these are rare cases, but they can happen. Because for this whole lecture, I want everyone to know the advantages and disadvantages, rather than a lot with vaccines. Usually we see people being very like black or white, and I don't want to do that. I want you guys to have a good understanding of the good and bad of vaccines. So an objective view. Another type of whole agent vaccines are inactivated killed vaccines. This is when you take the pathogen and you inactivate it or kill it. So it's unable to replicate in the body, but it can still mount an immune response, which is what we want from a vaccine. And they mostly induce humoral immunity. This is the one that has to do with forming antibodies. The advantage is they're safer than live vaccines because they cannot cause infections they can't replicate. The disadvantage with these vaccines is you have to keep giving booster doses. There's a lot of booster doses because they cannot replicate. So you need your body to create memory cells. And examples of this are Dr. Salk's polio inactivated killed vaccine, the hepatitis A virus vaccine, and then there's there's different types of the influenza vaccines. They keep coming out with them, but the influenza vaccine is an example of an inactivated killed vaccine. So those were whole agent vaccines, and there was attenuated and inactivated. Now we're going to talk about fractions of the agent. These are all inactivated, meaning they're basically all dead, but they're fragments of the pathogen, and these antigenic fragments or fragments of the pathogen will mount an immune response in someone. So examples of these are toxoid vaccines, which are when you inject inactivated toxin in a person so that if they ever are exposed to the toxin in their lifetime, they won't have the disease, so you won't get tetanus or diphtheria. These are part of the DTaP vaccine, which is a vaccine that consists of tetanus, diphtheria, and pertussis. There's protein subunit vaccines. These are vaccines where you take certain proteins of the viruses and you create vaccines. So pertussis, which causes whooping cough, hepatitis B affects the liver, human papillomavirus. They're all scientists figured out certain protein subunits of these viruses that will mount an immune response. So that's what we use in the vaccine. Polysaccharide vaccines are when we take, remember, polysaccharides are sugars that mount an immune response in an individual, and we use them as vaccines. So pneumococcal pneumonia, meningococcal disease, Haemophilus influenza type B, meningitis. Anytime you see the word meningitis, this is very deadly. It's not something you want to get. It affects the meninges or coverings in the brain area. And then DNA vaccines, specifically nucleic acid vaccines, they're still experimental. This is when we extract DNA or RNA from pathogens, whether that is bacteria or viruses, and we use that to mount an immune response. So they're still studying this and seeing how effective it is. So these are types of fractions. Childhood immunization. So when babies are first born, they have a high amount of IgG. Remember, IgG is an antibody that they get from their mom. So in the beginning of a baby's life, they have a high amount of IgG, and the IgG over that year gets catabolized or broken down as the baby's immunity builds up. So their immunity becomes low as they slowly start developing their own IgG. So there's the maternal IgG, and then the baby is slowly developing their own antibodies. And IgG is a great antibody that protects you against certain pathogens. Remember, antibodies can cause phagocytosis, neutralization, agglutination. And so childhood immunization is to help the children develop an immune response so that if they're ever exposed to a deadly pathogen, they're ready to target it. And just 2 more points. So federal law requires that vaccine information statements are given to parents or guardians when children get vaccinated. There have been a lot of fear of side effects with vaccines, as you've all heard about, but I want to mention that vaccination has dramatically reduced incidences of disease. And when we see a drop in vaccination or immunization, sometimes people interchange these words, vaccination and immunization, but when we see a drop, we see a lot of disease outbreaks in certain areas. So they are a very important part of our health. And then with adult immunization, there's certain boosters that we take for some vaccines to keep building memory in case we're exposed to it. Some specific jobs require more vaccines. So there's the general vaccines that everyone's supposed to take, and then there's vaccines that you, in addition, have to take if you have certain jobs. If you're a teacher, if you're a professor, if you work in the army, you take certain vaccines, additional vaccines. And there are travel vaccines. So if you travel to certain countries, you may be recommended to take a vaccine that you maybe would not have taken if you're just staying in the United States. So I just took this picture from the CDC's website. This is the recommended vaccine schedule for this year for children. So from birth to 4 to 6 years old. So if you can see at birth, actually the day the baby is born, you're asked if you want to give baby a hepatitis B vaccine, and then rotavirus vaccine, DTaP vaccine, hepatitis, polio vaccines, so just certain types of vaccines. And you could see, like, for example, the MMR vaccine you don't take until the baby is 1 year old. So certain vaccines, this is why babies here, so if you think of babies under 1, if we use measles as an example, they cannot get the vaccine for measles, mumps, rubella, which is all a vaccine until they're 1. So here we're depending on herd immunity to protect these babies until they can get vaccinated. So this is where herd immunity plays a role. And so I'm not going to go into the detail of every disease. I just put these tables up for you guys if you want to read about them. But there have been vaccines that have helped a lot with bacterial diseases in humans. So Haemophilus influenza type B meningitis is caused by bacteria, and the vaccine is created from taking the sugar from the bacteria and creating the vaccine. And remember that this causes meningitis, which it causes an infection in the meninges or coverings of the brain and spinal cord. So it's very serious. Meningococcal meningitis. We take sugar from Neisseria meningitis, which is the bacteria, and we help create a vaccine with that. Pneumococcal pneumonia is a lung infection that we also have the PCV vaccine. Tetanus, diphtheria, and pertussis. All of these are bacteria that are bacterial diseases. So it's the toxin that the bacteria produce that causes, like, for example, tetanus and diphtheria. So this is where we give the vaccine so that if you're ever exposed to tetanus, for example, you won't get muscle spasms. And then viral diseases. Vaccines have played a huge role in viral diseases. As all of you guys know, with the COVID-19 right now outbreak, viruses are very hard to treat, especially because antivirals are hard to create and they're not very effective a lot of times. So we have vaccines. We have chickenpox vaccine, which causes like a bad rash, itching rash on the body. The herpes zoster vaccine for shingles, which is a very, very, very painful rash. Hepatitis A vaccine for liver infection, hepatitis B. There are different viruses cause different hepatitis. They all affect the liver, some worse than others. The human papilloma virus can cause many different things. The main feature is warts on the body, painful, and then influenza. More viral diseases, measles, mumps, rubella, rotavirus, polio. So all of these diseases we've created vaccines for that have been very effective. I want to say no vaccine is 100% effective. This is the same idea of saying chemotherapy is 100% effective. Chemotherapy is not 100% effective, neither are vaccines, but it's always outweighing the good and bad. That's with any medication in the world. So on very rare occasions, vaccines can cause a disease. I wish we were in class, we would discuss this. We always discuss this, but I just want to say that, again, there is no right and wrong with vaccines. It's not black and white. Vaccines have side effects and they are great at the same time. It's the same thing when you think of chemotherapy and different medications we use to treat cancer. So with everything in the world and regarding the world of health, you always try to weigh good versus bad. And there's been no medical or scientific proof that the MMR vaccine, this is the one that's been linked to autism, actually causes autism. So if you read more in detail about all the stories with the MMR vaccine and autism, it's not true. Vaccine right now are the safest and most effective means of preventing infectious disease in children, especially things that can be very serious in children. Now we're going to talk about immunoassays. So we finished with vaccines. The other part of learning about your immune system, now that we talked about vaccines, is how can we use components of the immune system that we learned about to create really great diagnostic tests that we can use in hospitals and research settings. So immunoassays are lab tests that use antibody-antigen interactions to detect given antigens or antibodies in a sample, meaning they're basically lab tests to detect, does someone have antibodies for coronavirus? Does someone have coronavirus in them? So you're using different immunoassays. Immunoassays are many different tests, and we're looking at certain tests to look at antibodies or antigens. You're trying to determine if someone, for example, has an infection. That's a certain immunoassay. You can determine pregnancy tests. That's an example of an immunoassay test. Any test that uses antibodies, antigen interaction to figure out something is called an immunoassay test. And it's used to determine the presence and or concentration of a component through the use of an antibody or antigen. I'm going to talk about this picture in the next example. So the disease syphilis is caused by bacteria. And so the bacteria is called Treponema, and it causes syphilis. Syphilis is an STD. It's a sexually transmitted disease. You can use an immunoassay to determine if someone has this infection, if someone has syphilis. And there's two ways. You can bind known antibodies and identify your unknown pathogen. So you can have antibodies that are specific to the bacteria that cause syphilis, and here's the bacteria that cause syphilis. If they bind, then you know that that individual has syphilis. Or you can take a blood sample from someone and see, do they have the antibodies that actually bind the pathogen? So you take antibodies from the patient's serum or the fluid component of their blood, and you mix it with the actual organism that causes syphilis or causes whatever disease you're studying. And if you see interaction, then they do have the disease indeed or infection. So with immunoassays, you can use known antibodies with unknown pathogens or known pathogens with unknown antibodies found in someone's blood. So there's many immunoassays exist, but they all go on the same basis of, does an antibody bind an antigen? And you're going to determine that by different things. The immunoassays we're going to talk about are agglutination reactions, fluorescence antibody tests, enzyme-linked immunosorbent assay or ELISA, and Western blot. These 4 immunoassays or tests are lab tests that we do that test, did an antibody bind an antigen, and they all visualize this in different ways. So just to give a summary, in agglutination reactions, so these are all things you do in lab. If the antibody and the antigen bind, you get agglutination, you get clumping. And I'm going to tell you, you physically see clumping. I'll show you a picture of that in a second. With the fluorescence antibody test, from the name, if the antibody binds the antigen, you get fluorescence. With enzyme-linked immunosorbent assay ELISA, if the antigen binds the antibody, you get a color change. With the Western blot, if the antibody binds the antigen, you see a band on blot, which I'll talk about what that is. So all of these are different ways of visualizing, did the antigen bind the antibody? An example of this is if you take, for example, a blood sample from someone and you're trying to determine, do they have antibodies for SARS-CoV-2, so for coronavirus, and you could do any, if in theory all these tests were established. I know ELISA has been established for coronavirus, but if they were all established, if the person does have the antibody for the virus is the antigen, the antibody is the antibody. If they bind in agglutination reactions, you would see agglutination. In fluorescence antibody tests, you would see fluorescence. In ELISA, you would see a color change in the well that you do the experiment. In the Western blot, you would see a band if you had antibody antigen bind. So with all of these tests, you can identify either an unknown pathogen or an unknown antibody. And we'll start with agglutination reactions. So agglutination reactions have to do with agglutination. If an antibody binds an antigen, you'll get visible aggregates or clumps. In, like, if you did the reaction on a piece of paper, you see clumping. And there's direct agglutination and indirect or passive agglutination. With direct agglutination reactions, we can use these tests in lab to detect if a patient has antibodies to a particular antigen. So you have unknown antibodies in a person, and you're going to test known antigens to see what the antibody binds to. So you have an antibody in someone, you know that there's an antibody, you just don't know what it's for. So you're going to take that unknown antibody and you're going to mix it with different pathogens that you suspect. Wherever you see clumping or agglutination is where you know that that antibody is for that specific antigen. And that's an agglutination reaction. So you have different known antigens, which are pathogens, on a microtiter dish, so dish with different wells, and then you add the patient's serum, and wherever you see agglutination, here's a good picture of agglutination or clumping, you know that that's what they have. And again, the direct agglutination reactions say that the antibody binds the antigen directly, and you have a known antigen and unknown antibody, and whenever you see clumping, it means that they match each other. So if the patient's serum has antibodies to a known antigen, you get the clumping. And the outcome of direct agglutination reactions is you determine the identity of unknown antibodies in a patient. So a patient comes to you, take a blood sample, you see antibodies, you're not sure what they're for, you can do this test and you can determine what type of, for example, bacterial infection they have. And we also use direct agglutination reactions with blood. So blood antibody and antigens. Remember, when antibody and antigen bind to each other, you see clumping. So this is how we determine someone's blood type. You take their blood and you mix it with different antigens, and whenever you see clumping is where you can tell the blood type. So direct reactions used in blood typing, and clumping is a blood type. So for example, this person has antibody A, and antibody A binds to antigen A, and so you get clumping. Indirect agglutination reactions are also called passive reactions. It's very similar, but there is a middleman in lab if you're actually doing it. So here we're doing with indirect agglutination reaction is you're using latex beads in kind of in the middle between this reaction of antibody antigen. So with the indirect or passive or latex agglutination reactions, you can assay for an unknown antibody. So if someone has an unknown antibody in their blood, for example, you can use this test to determine the known antigen. So the known antigen, you test different known antigens with the unknown antibody, and they're bound to latex beads. You take different antigens, different bugs, and you bind them to latex beads. And if they bind to the unknown antibody, that's the antibody the person has, you can also assay for an unknown antigen. You take known antibodies that have latex beads bonded to them, and wherever they bind or where you see agglutination, you can determine the bacterial antigen. So again, here you have the antigen or the antibody bound to latex beads, and the latex beads help you see the reaction stronger. So when we see latex beads, that's considered an indirect agglutination reaction, whereas the one with no latex beads, where the antigen binds the antibody and you get clumping, that's considered a direct agglutination reaction. So the take-home message from agglutination reactions is that with agglutination reactions, you can determine if someone has the unknown antibody or unknown antigen by if you see clumping. So if the unknown antibody binds to the known antigen, you get clumping, you can determine what type of infection someone has, for example. The fluorescence antibody test, when antibody binds antigen, we see fluorescence. So with this test, we use antibodies tagged with fluorescent molecules, and when the antibodies, so if you have your antibody as a V or as a Y, because they mostly look like a Y, and you have a fluorescent molecule on it, every time the antibody binds the antigen, you get fluorescence. There's direct and indirect fluorescence antibody tests. Direct fluorescent antibody tests can direct an unknown organism from a clinical specimen directly. So when you see the antibody with the fluorescence directly bind the antigen, you get fluorescence. That's their direct one. If you use a middleman or another antibody, that's an indirect fluorescence antibody test. And remember, whenever you're looking at fluorescence, you need a fluorescent microscope. Okay, so for the direct fluorescence antibody test, it uses antibodies tagged with fluorescent molecules. Whenever these antibodies bind to their proper antigen, you get fluorescence. So if someone, let's say for example, you suspect someone has strep throat, so you send a sample of you send a patient throat sample to the lab, You're not sure what it is. What the lab can do is they have streptococcus antibodies, fluorescent streptococcus antibodies that they can mix with the patient's sample. The patient's sample is considered the antigen. If they bind to each other, if the patient sample is actually strep and the fluorescent antibodies, strep antibodies bind to the sample, and you go look at it under the microscope and you see fluorescence, you know that they have an infection. Now, if the patient's sample had like another virus, streptococcus is bacteria, but let's say it had a virus that causes a sore throat, the antibodies will not bind, so you'll not get fluorescence. So we can do this for clinical tests. The indirect fluorescence antibody tests is used to detect a specific antibody in serum. So you're going to take known antibodies and stick them to a slide, and then you're going to add the patient's serum because you're not sure what antibodies they have. So let's say you're testing for a specific bug, a specific pathogen. So here's your pathogen, and you add the antibodies from the patient's serum and lab. So you take a blood sample, they bind to each other, then you add another antibody. This is why this is indirect, because the fluorescence is attached to another antibody, and this antibody attaches to the original antibody. This antibody is called anti-human immune serum globulin, and it reacts with any antibody. So it sticks to this antibody that's stuck to your antigen or pathogen, and then you get fluorescence. And here in this example, we're looking at spirochete. So this is a spirochete that causes a disease, and you can determine that that patient has a disease. So you're adding, remember, another antibody that has fluorescence on it. So that was the fluorescence antibody testing. So there was direct and indirect. Now we're going to talk about ELISA, which stands for enzyme-linked immunosorbent assay. So this is used to detect either antibodies or antigens in a patient's serum or the fluid portion of their blood. And this is what they're doing with one of the coronavirus testing, is an ELISA test. So you use antibodies tagged with an enzyme that will catalyze a color change. So with ELISA, when your antibody binds the antigen, you get a color change. So if you look at, this is called a microtiter plate, it has different wells. Every time we see a color change, we know that the antibody and the antigen bound. So let's say this has 96 wells. Let's say you took 96 patients and you were trying to determine if they're infected with SARS-CoV-2. Wherever you see a color change, meaning that the antibody bind the antigen, they have the infection. So it's performed in a microtiter plate. A positive reaction produces a color change. So for example, this well right here, I would say that the patient doesn't have an infection because it stayed clear. Where we saw a color change, they do. And there's a direct ELISA, which detects antigens, and an indirect ELISA, which detects antibodies. With the direct ELISA, you're detecting antigens using a known antibody. So on your wells, it's coated with antibodies. So you have antibodies here. For example, let's use the pregnancy test as an example. The pregnancy test, you cannot see this, but on that strip, there are antibodies there that will detect the HCG hormone. So if urine has the HCG hormone, it'll bind to the antibodies that are already on the strip, and you'll get a color change. That's an example of a direct ELISA. And if no hormone is present, the antigen is not, you will not see a color change. So that's a direct ELISA. It detects antigens using the known antibody, and whenever the antigen binds the antibody, such as the pregnancy hormone HCG binding the HCG antibody, you get a color change. And remember, with ELISAs, you add another antibody with an enzyme on it that catalyzes a color change. So pregnancy tests are an example of a direct ELISA reaction. With indirect ELISA reactions, you're detecting antibodies in a sample using an antigen. So I assume that this is one of the tests that we're using with coronavirus. So you have wells that are coated with different parts of the coronavirus, let's say the spikes. So when we look at the virus, it has spikes that help it get infected. respiratory cells. So when we look at the wells that they're using, they have them coated with different parts of the SARS-CoV-2 virus. And you take a sample from a patient and you, I was going to say squirt it, you pipette it into these wells. If they have the antibodies for coronavirus, it will bind to your bound antigen, which is some part of coronavirus. And then you'll add another antibody with the enzyme that catalyzes a color change. And then there you'll get the color change and you'll know that the person actually has an infection. So screening for HIV is done with this. The reason is that HIV antigens are, it's easier to look for HIV antibodies than HIV antigens because HIV antigens hide in cells. So with it's easier to have wells that are coated with different parts of the HIV virus, and then you take a blood sample from someone and you pipette it. And so that's why with HIV, we prefer to use an indirect ELISA. But you can get occasional false positives. False positives is when you see a color change and the person actually does not have the reaction. So we usually do more confirmatory tests. The last immunological test I'm going to talk about is the Western blot. The Western blot is usually a test we do to confirm that, like let's say if someone did an indirect ELISA and we saw a color change, so we suspect that they have an HIV infection, you would then do a Western blot, which is another immunoassay. Remember, all immunoassays are looking for, does the antibody bind the antigen, and what am I seeing as a result? Are you seeing agglutination? Are you seeing fluorescence? Are you seeing color change in a well? With the Western blot, you're going to see a band on a blot. A blot is like a piece of paper. I'm going to explain this as easy as possible because you really can only understand if you do the experiment. A blot is a filter piece of paper, and if your antibody and antigen bind to each other, you're going to see a band on it. That's the easiest way to explain it. So what you do is you run the person's sample and then you add antibody on your blot. And if you see a band, then you know the person has that antibody, has that antigen or pathogen. So it's a direct method. It's not indirect. It's a very direct method. It's a great experiment to do to identify if someone has a protein using an antibody for that protein. And so we use known antibodies tagged with an enzyme to identify the presence of an unknown protein in a patient, or we use this a lot in research. And if you see the band, it means that a person does actually have that protein. So for example, if we look at the HIV virus, or you could think of the coronavirus, when you look at the virus structure, the virus has different proteins. Spikes are proteins. It has the capsid, that's a protein. You can use these components of the virus to create antibodies that will bind to them. So what you do is you take a patient's sample and you could do a Western blot, and then you take antibodies that will target the spike proteins of coronavirus or the spike proteins of HIV, and you pipette them on your blot. And if you see a band, then you know that they actually have that protein that indicates a viral infection. So this is used to confirm ELISA results. And again, it's a confirmatory test for HIV. Finally, we're going to end this lecture talking about monoclonal antibody therapy. So in the adaptive immunity, we talked about humoral immunity. Humoral immunity has to do with making B cells that make antibodies, and antibodies are great proteins because remember, antibodies target cells that are infected, and antibodies can lead to different things. They can lead to neutralization, they can lead to phagocytosis, they can do a lot of things, but basically antibodies are protecting you against whatever infection you have. B cells make antibodies. B cells are a type of white blood cell. So with monoclonal antibody therapy, you have someone who has a certain disorder or disease, and to make it as simple as possible, you're giving them antibodies so that those antibodies can target certain disorders. So how you do it is you isolate and immortalize B cell from a patient. So if you have a patient that fought a certain infection, you can take B cells from them because you already know that they're producing antibodies against that infection. You can clone it in lab, meaning make many copies of that B cell, and then you cause that B cell to produce one type of antibody that recognizes one antigen. So that's a monoclonal antibody. And then you use that antibody, you can use it in medication, you can inject it in patients who are sick so that it helps them fight whatever it is they're fighting, because we've used this with various things. So monoclonal antibodies attack antigens. We've used monoclonal antibody therapy in cancer and immunologic disorders, so rheumatoid arthritis, asthma, other things. Examples, which I think they have a lot of TV commercials, so some of you guys may have heard of these. So Herceptin is used for breast cancer, Humira for rheumatoid arthritis, and with Humira, for example, you're giving that person antibodies. That's what a Humira is. And the antibodies neutralize the cytokine or the signaling molecule that causes inflammation. Therefore, you get less inflammation on joints, which is a great thing for patients with rheumatoid arthritis. Zolar for persistent asthma. So you prevent the binding of the IgE antibody on mast cells and basophils by neutralizing it. And if it doesn't bind, then you won't get so much release of histamine and all the other different components of IgE that are released when they bind the white blood cells. So here, for example, you have a cancer cell and you can get the monoclonal antibodies to attack the cancer cell. And by attacking it, they can affect this cancer cell in different ways. And then here, if we look, so you have so Herceptin is used for breast cancer and it's a monoclonal antibody. So HER2 receptors send signals telling cells to grow and divide. When you give the medication stops the receptors from telling the cell to keep dividing and dividing. So it's more complicated than that, but what I want you guys to get from this is with monoclonal antibody therapy, we're taking B cells and we're trying to get them to make us one type of antibody in lab and then producing a lot of that antibody and using that in medication to target different types of infections. So this was the immunological applications lecture. And again, we talked about vaccines, different types of vaccines, and different types of immunoassays to know how to categorize whole agent vaccines from subunit fraction vaccines, and then know the different immunoassays we talked about. So agglutination reactions, fluorescence antibody tests, ELISA, Western blot, and no direct versus indirect, and where examples fall of each. So good luck with this.